So, we talked about the basic structure of DNA and RNA nucleotides, right? They're nucleic acids. They're composed of three different things: a phosphate group, a sugar, and an nitrogenous base. We learned that the sugar carbons are numbered in a specific way. And we learned that nucleotides, they're added to the 3' ends via dehydration synthesis to create something called a phosphodiester bond. Okay. So, the last thing I'll say before we turn this over is that the function of nucleic acids that we'll dig into more during Unit 6, but the major function is that it stores information that is necessary for the proper functioning and regulation of a cell and the expression of traits in an organism. Helps with regulation and functioning of a cell, but also the expression of traits in an organism. And the last part is that usually the info or the information stored is used to create specific proteins. All right. Back to your biology class, regular bio, you probably saw something like this. All right. DNA is transcribed into RNA and translated into a protein. Okay. We're going to get much more specific in Unit 6, but that's the general flow of information. DNA creates proteins. All right. Is anybody still writing? Turn it over. So for this particular unit, the reason we're talking about DNA is you have to be familiar with the structure. So we have two organic molecule models up here. This one is DNA, and this one right here is RNA. Talk about DNA first. Okay, and for context, a more zoomed out structure of DNA. Is known as a double helix. Okay. And we are specifically zoomed in So we're looking at just a few base pairs in the DNA molecule. Okay, so what you need to know is that this molecule, that's the midway point. Okay, so through the middle, we have two strands that are held together by a hydrogen bond. Two strands are connected in the middle by hydrogen bonds, but the strands on their own are connected via phosphodiester bonds. Okay, so this phosphate right here, this phosphate group is involved in two ester bonds. This right here, that's an ester bond. So there are two ester bonds connected by the phosphate. Okay. So remember, nucleic acids are made up of nucleotides. So this is one nucleotide right here. Has a phosphate, a sugar, and a nitrogenous base. Okay. And if you notice, there are four different types of nitrogenous bases. Probably heard of these before, but you have a cytosine, which is the nitrogenous base of the one I've circled. You have a thymine, you have guanine and adenine. Okay. And this is where you've seen a G connects with a C, right? If you remember base pairing rules, and a T connects with an A. All right. So, if you look in the hydrogen bond area, let's pick this one right here. So the bond between thymine and adenine. So, nitrogen is connected to a hydrogen right here. Which of those two atoms is more electronegative, nitrogen or hydrogen? Nitrogen is significantly more. So although the partial charges aren't indicated, that'd be a partial negative on the nitrogen and a partial positive on the hydrogen, which is why we have a hydrogen bond forming with the nitrogen over here, which has a partial negative charge. Because what is this nitrogen connected to? What is this nitrogen connected to in the molecule? Carbons, right? 2 carbons. Carbons are significantly less electronegative, which is why nitrogen has a negative charge. Okay, so that's when we get a hydrogen bond. Okay. You'll also need to be familiar with the fact that we have directionality in the molecule. So we indicated, we'll go back to our nucleotide that we circled, that we number our carbons in a particular way. So we have the 1' carbon, 2', 3', 4', and 5'. Right? So the 5' has the phosphate, 3' has the OH. Okay? What do you notice about both sides? So this side and this side, specifically with regard to the 5' and 3'. What do we notice? Yeah.
The 3' end and the 5' end are not like opposites of each other, so like that makes DNA in like anti.
Yeah. So she's pointing out that if you notice at the bottom of the left-hand strand, right, we have the 3' hanging off. But on the other side, it's the opposite, the 5'. And so we call this anti-parallel. Right, so one strand is running in one direction, and the other is going the other direction. Okay. So the last thing you need to know about DNA structure is that there are 2 types of nucleotides. 2 types of nucleotides. You have A and G's and T and C's. So you have adenine, guanine, and thymine and cytosine. Talk to your neighbor real quick. When you look at their structure, here's guanine, for example, and cytosine. When you look at the structure of those 2 and these 2, how are they similar? Go ahead and look real quick. How are they similar to one another?
Oh.
All right. So, Evan, what are your table finds? How is adenine and guanine similar? How are thymine and cytosine similar?
Guanine and adenine have
So you're talking about this right here? Connected to what? So he's saying he noticed that adenine and guanine have this little pentagon. Keep in mind this whole thing is your base. Right? We have base, sugar, and phosphate. So the base in A and G, is it bigger or smaller than the base in T and C? So adenine and guanine, right, their base, is it bigger or smaller than the base of a cytosine and a thymine? It's bigger, right? It's huge, right? So it's got 2 rings instead of 1 with its base. And so we call these double ring bases, double ring bases. And T and C are single ring bases. Right? Hopefully you see why. One has 2 rings, the other one just has 1. The scientific description is that these are purines. They call these purines. T and C they call pyrimidines. Okay. So I would figure out a mnemonic of how to remember that A and G, those are the bigger ones, are called purines. For me, when I was in high school and college, A and G stood for all gold, being like pure gold. That works for you, great. But A and G in my mind stood for all gold, which gold is pure, or that when it's really pure, it's valuable. And so that's how I remember that those were the purines. And gold costs a lot of money, and so it's the bigger one. So that was just one way that I remembered it, but I would just be familiar that they are purines and they're double rings. They're different from these. Okay. Questions? Yeah.
Where does it show that T and C are single rings?
Great. Good question. So if you look at cytosine and our thymine, all right, look at its nitrogenous base. So I'm going to circle the base for you. Right? The ring I'm referring to is that ring. Right? So cytosine has 1 ring, but adenine has 2 rings. Right? Any other questions? Okay. So with RNA, we're not going to spend very much time because it's very similar, except it just has 1 strand. But the differences that I point out, so they already have them labeled for you. So this top nucleotide is adenine, guanine, cytosine, and uracil. The major difference that you're seeing is that there's no thymine in the RNA. It's uracil instead. I want you to turn to your neighbor. Specifically compare how are the sugars of a DNA molecule different from the sugars of an RNA molecule. So just the sugar component. Turn to your neighbor, how is it different between the 2 nucleic acids? So help you out. They're in pink right here. That's a sugar from RNA. That's a sugar from DNA. How are those similar and different? All right. Angel, I think I overheard What difference did you see between the two of them?
In RNA, there's 2
In RNA, there are 2 OHs, but in DNA, how many OHs are there? Specifically, what carbon in the DNA molecule is missing an OH? What carbon in the DNA sugar is missing an OH? Here's the numbering again right here. 1, 2, 3, 4, 5. Which carbon is missing the OH? What carbon is missing H? Okay. Does anybody know what DNA stands for? Nucleic. Oxy is because it's missing an oxygen on the, so it's missing. Okay. DNA from RNA, which RNA stands for just. Because it, all right, the last thing I'll say about RNA is to classify uracil as a pyrimidine. A pyrimidine, T, C, and U. G and C, T and A. We know that in both of these cases, let's take DNA for example. Serine or pyrimidine. C is a pyrimidine because it creates a symmetrical molecule. Right? Because it's one. All right. I will stop talking. Any last questions before we take a short brain break?